Welcome back, my dear students. Today we are going to explore one of the most fascinating features of Porifera or sponge, the canal system. It's like the sponge's very own plumbing network where water constantly flows in and out, bringing food and oxygen and carrying away metabolic waste. This system not only keeps the sponge alive but also makes it one of nature's most efficient filter feeders. So let's dive in and discover how this amazing water current system works inside these simple yet smart animals. Canal system in sponge. Introduction. The canal system is a unique feature of the phylum Porifera or sponge. It was discovered by Robert E. Grant in 1825. The body of a sponge has minute pores or ostia through which water enters. A large opening is there which is known as osculum, through which water leaves. There are so many canals like radial canal, incurrent canal, or flagellated chambers. And there is a central cavity. This is the cavity known as spongocoel. So water flows in one specific direction, and that is unidirectional flow from ostia to canals to spongocoel, and finally it moves out through the osculum. Functions of the canal system. The canal system play four major functions like nutrition, respiration, excretion and reproduction. Nutrition. Sponges are filter feeder, so they have choanocytes or collar cells which capture food particles from the water as they are filter feeder. But the digestion occurs inside the amoebocytes, and undigested food leaves through the osculum. Respiration. Exchange of gases occurs by means of simple diffusion. Here oxygen from water enters the cells and carbon dioxide diffuses out. Excretion. Ammonia or NH3 and other metabolic waste products diffuse out into the water as it passes through the amoeba, passes through the body. Reproduction. During sexual reproduction, sperms release from choanocytes and travel through the canal system and fuse with ova inside the sponge body. Types of canal system. The canal system in sponge show a gradual increase in complexity. There are basically four main types of canal system: Asconoid type, Syconoid type, Leuconoid type and Rhagon type. Here the Asconoid type is the most simplest type of canal system, and Leuconoid type is the most complex type of canal system, and Syconoid type is act as a bridge between the Asconoid and Leuconoid type of canal system, and Rhagon is a canal system found in some larval sponges. Asconoid type, it is the simplest and most primitive type of canal system. It is found in the animals like Leucosolinia and in the Olinther stage of Clathrina. The body is basically vase shaped. It's a flower vase like structure, and it is radially symmetrical. The body wall is thin here, and it encloses a spongocoel or atrium. The choanocytes line the spongocoel directly. Here the ostia or pores open directly into the spongocoel through porocytes. Here water flow from ostia to spongocoel and then osculum. Water current is created by the beating of flagella of choanocytes. This is the Asconoid type of canal system. Syconoid type, it represents an intermediate or transitional stage between amoeboid, Asconoid and Leuconoid type of canal system. It is found in Sycon or Scypha and Grantia. Their body wall is folded into finger-like projections called radial canal, and it is lined by the layer of choanocytes. Spongocoel is not lined by choanocytes, but there are pinacocyte cells. Type of Syconoid canal system. There are basically three types of Syconoid canal system. Simple Sycon type, it is found in Grantia and Leucandra. The radial canal shears are separate and open directly into spongocoel. There is no distinct incurrent canals are formed. So water from exterior moves into the interior incurrent pore, and then it comes into spongocoel, and then it comes into osculum, and then water moves out. Complex Sycon type, it is found in Scypha and Sycon. Incurrent canals and radial canals are present alternately. Here incurrent canals open to the exterior by means of ostia, and radial canals are connected to incurrent canal through prosopyle and open into the spongocoel via apopiles. The water flow as directed ostia into incurrent canal, then prosopyle into radial canal, then apopyle, then spongocoel and osculum. Sycon type with cortex, it is found in Grantia, Grantiopsis, Uti, etc. A thick cortex layer covers the body. An incurrent canal pass irregularly through the cortex before reaching radial canal. It may contain large subdermal spaces. Syconoid type. The Syconoid type basically represents a stage between the simplest Asconoid type of canal system and the more complex types. It is the first step towards complexity. The body wall here forms a finger-like projections called the radial canal at regular interval, and it increasing the complexity compared to the Asconoid type. The choanocytes, flagellated cells, are limited to these radial canals, which are extension of the spongocoel or the main body cavity. Here the radial canals are lined with choanocytes and responsible for water movement and food collection. In this type of canal system, water enters through dermal ostia or external pore and flows through the incurrent canal to the radial canal. Incurrent canals from tubular spaces between the radial canals and are lined by flat cells or pinacocytes. The walls between radial and incurrent canals are pierced by small pores called prosopiles for water flow. Water then moves into the spongocoel and exits via the osculum. The Syconoid system is found in calcareous sponges like Sycon and Grantia. This is the Syconoid type of canal system. This is the early stage without cortex, and this is the final Syconoid type canal system with cortex. Here the dermal pore, through the dermal pore water comes in and it moves into the incurrent canal, then it goes into prosopile, then into radial canal, then into internal ostia or apopyle, then spongocoel, and then through osculum water moves out. Leuconoid type of canal system, the most complex and advanced type. It is found in demospongi such as Spongilla, Cliona and Euspongia. There are flagellated chambers replace the radial canals. Dermal pores open into subdermal spaces leading to incurrent canal. Then water moves into flagellated chamber and then moves into excurrent canal and then moves out by means of osculum. The spongocoel is much reduced. Choanocytes line only the flagellated chamber, not the spongocoel. Types of leuconoid canal system: uripilus type. It is the simplest leuconoid form. Flagellated chambers open directly into the excurrent canal. It is found in Placina, Jetila, etc. Aphodal type, a narrow canal or aphodus connects the chamber with excurrent canal, found in Geodia and Stilata. But the diplodal type, it is found in both prosodus between incurrent canal and chamber, and aphodus between radial and excurrent canal. Between chamber and excurrent canal are present. Both the prosodus and aphodus are present. Advantages of leucon type: efficient water circulation is performed by this type of canal system. It is better for food capturing and respiration. It increases body size and complexity. Leuconoid type. The leuconoid canal system develops from the Syconoid type due to outfolding of the choanocyte layers and thickening of the body wall. The choanocyte layer of the Syconoid radial canals folds into small rounded oval flagellated chambers. These chambers replace the elongated ones of the Syconoid type. In the leuconoid type of canal system, choanocytes are limited to these small flagellated chambers, and the space around the flagellated chambers is filled with the mesenchyme or gelatinous connective tissue. The spongocoel or central cavity is often reduced or absent, leading to an irregular or indefinite body form. Water flow system of this type of canal system includes: many incurrent canals bring water into the sponge through dermal pore or ostia. The water then flows through the prosopile into the flagellated chambers. From the flagellated chamber, water exits via apopiles into excurrent canal, which further merges to the large canal that lead to the larger opening or spongocoel. The surface of the sponge is covered with epidermal epithelium and pierced by many dermal pores and oscula. Now it is the last type of canal system, or Rhagon type. Here the conical body with the osculum, it's present at the top. This is the conical body and osculum is present at the top. Spongocoel is here. It is surrounded by oval flagellated chambers that open into it by wide apopiles. These are the apopiles. This thick mesenchyme with incurrent canal and subdermal spaces, they are found in the larval stage of demospongy. So the Rhagon type of canal system is found in the larval stages of demospongy. It is considered as a developing stage between the Syconoid and Leuconoid type of canal system. Here the direction of the water flow from the ostia, and water takes in, and through the osculum it moves out. So what is the mechanism of water circulation? Choanocytes line the flagellated chamber and beat their flagella continuously. This creates a water current that draws water in through ostia, passes it through canals and flagellated chambers, and then pushes it through the osculum. Prosopiles are small opening, they allow water to enter chambers, and apopiles are larger opening, they let water flow out. Water flow helps in feeding, respiration and excretion. So my dear students, today we explored the fascinating canal system of sponges, the unique water transport mechanism that keeps these simple animals alive and active. We learned how water enters through ostia, travels through canals and chambers, and finally exits through osculum, performing vital functions like feeding, respiration, excretion and reproduction along the way. From the simple Asconoid type to the highly complex Leuconoid type of canal system, this evolution shows how sponges become more efficient and more organized over time. Truly, the canal system is the lifetime of sponges, beautifully demonstrating that how even the simplest animals have evolved remarkable ways to sustain life. Thank you for watching the video. Keep exploring, keep questioning, and I will see you in the next class again. Thank you. Bye.